Mike, I want to bring up something that I mentioned to you maybe some time ago that I think is really serious. Um, I think we disagree, maybe not, but let's get it said. And I think that your Xenobots are the way to test it. So I know that I have sent you the Andrea Rolli Stu Kauffman paper, A Third Transition in Science. Our, let me just go through very briefly our argument.
Go for it.
Organisms are Kantian wholes, said Kant, where the parts exist formed by means of the whole. So you're a Kantian whole. You get to exist, you've got livers and kidneys. So you exist because you have them, but they exist because they're part of you. Now, the definition of a Kantian whole already allows us to talk about the function of a part. It's that, and I can say it precisely because I've said it lots of times, it is that subset of the causal properties of the part that sustains the whole. So for the heart, it's pumping blood, not making heart sounds. Which then leads to the next point. A given part, its function is a subset of its causal properties, but it might have other subsets of causal properties that are of use. And those are Darwinian pre-adaptations or exaptations, a la Verba and Gould. Now, intermediate and next step is the critical one. From one use of a thing, you cannot deduce it. You cannot deduce its other uses. So my beloved example is an engine block, and you can clearly use an engine block as a paperweight. And you can jury rig, and you can use the engine block with its rigid corners to crack open coconuts. So the critical step is from the use of an engine block as a paperweight, you cannot deduce its uses to crack open a coconut. Now, if that step is right and evolution has acceptations in them, and it does, they're not deductive, and therefore evolution cannot be deductive, and therefore there can be no entailing law. Then I want to make, that already tells us that evolution is not a deductive process. It's a continuous jury rigging process with no entailing law. And that means really complex things can come into existence without deduction. So hold that. Let me make the same claim about human invention. So you may or may not know that I came up with this TAP equation, which basically says you make things out of things by combining them. So the one example we'll do, the printing press is a recombination between movable type and a wine press. But what one is doing is you're using things for funny reasons and funny ways, and in general, you can't deduce it. So human innovation is also not deductive, and it shows up because in patents, it has to be creative over the original over the prior art, which means it's not obvious. And typically it's because it's not deduced. Now, if that's right, the notion that cognition is limited to deduction is wrong. All computation is algorithmic, and therefore it's deduction. Therefore, human mind and the evolution of the biosphere is not limited to deduction, which is more than Gödel's theorem, which says if you've got a formal system within it, you can have formally undecidable statements. This is beyond Gödel's statement. So that's a long preamble to something I fell in love with your Xenobots. You take some skin tissue, some frogs or something, and the cells organize themselves and do weird things. It's just stunning that you did that. They seem like superb places to look, Michael, and I've gotten now to my real question. Can you look at, or, me, or Kate, or Joe Schmolowitz, look at your Xenobots and establishes that cells are using subsets of causal properties to achieve new functions and new functional integration in the Xenobot that are not the causal subsets of the cells that are normally used. In other words, are they jury rigging themselves so they get to exist? There's a final paper. I'm writing a paper with Andrea Rowley and some other people, including some soil scientists. And the basic idea is life will find a way. So I'm really excited about this theme. If every part can get used in indefinitely many ways, how many parts do you have to have? So they'll just manage to find a collective way of enabling one another. And so they get to exist with one another. And so the paper will be Life Will Find a Way. And are your Xenobots a stunning example of it? There, I've asked it.
Nice. Well, first of all, I kept waiting for you said we disagree, and I kept waiting for the thing that we're supposed to disagree on. I don't disagree with anything you just said. In fact,
great. Okay.
no, I agree with that completely, with a couple of twists that we can talk about. But let's just talk about the bots for a second. First of all, I don't know if you've seen so beyond the Xenobots, there have been the Anthrobots now. Have you seen those? Have you heard of this? So
I've heard of, but I don't know. Do you know, Kate? But anyway, what are they?
so let me tell.
Made out of human lung tissue.
Yeah, so let me tell you how that came about. So when we first made Xenobots, some people said, well, this has got to be a frog specific embryonic thing. These are animal caps. This is what frog embryos might do. It's a very kind of niche specific thing. I said, okay, what's the furthest we can get from amphibian embryos? Well, that would be an adult human. And so because I didn't believe for a second that this was some specific, frog.
This, of course, not. What a stupid belief.
But well, lots of people said it. We caught all kinds of heat for calling them anything other than animal caps because developmental biologists have been looking at animal caps for decades. So okay, so what we did was and this was the work of Gizem Gomushka, who's a PhD student in my lab, and a few other collaborators. What we did was we took tracheal epithelial cells taken from donors, and we put them in a different environment where they did exactly the same thing as the Xenobots do, which is they rebooted their multicellularity and they created Anthrobots. If I show you an Anthrobot, I could show you a video of it. It looks like some sort of unicellular prehistoric thing running around. They have autonomous motion. They have all kinds of interesting behaviors. But here's one other thing they do that's kind of interesting. If you take a petri dish and you seed a lawn of human peripheral neurons, okay, so there's a nice neural network growing on the bottom of this dish. You take a scalpel and you put a big scratch through it. So, a wound assay. So you put a big scratch through it, you throw some Anthrobots in, they run around, they can settle in the wound. And 4 days later, if you pick them up, what you'll notice is that what they've meanwhile done is taken the two ends of the scratch and healed them together. They heal these neural wounds. Now, who would have thought that your tracheal cells, which sit there quietly for decades in your airway, will actually have the capacity to run around, have autonomous motion, and by the way, heal peripheral nerve wounds when they come across them? Quite new. And so that's published. The thing that isn't published that I can just tell you a little bit about now, we're gonna preprint it probably in a couple of weeks, if not earlier. We've looked at transcriptomes both for Xenobots and for Anthrobots. Okay, because one question you might ask is, well, what genes do these things express? Because the conventional story is, well, your genome tells you what your anatomy and what your lifestyle is going to be. That's the conventional story. We looked at both Anthrobots and Xenobots, and we found thousands of new genes that the original source material does not express. Okay, these are things picked out of the genome, and once the preprint comes out, you can see all the different categories, all kinds of amazing things that are perhaps interesting in their new lifestyle that the native material does not express. So what you have here now, remember, in all of these things, there are no drugs, there are no synthetic circuits, there are no transgenes, there's no genomic editing. These are completely wild type cells, which have now reassembled themselves into a new form factor, and now they have some novel behaviors. Now, what we have not yet published, this will come later in the year, is, and this I think is important, because as you said, Stu, yes, they reconfigure themselves into new anatomies and new behaviors. But what we haven't shown yet is any of the stuff that I'm really interested in, which is goal-directed behaviors. So we haven't published that yet. What we show right now people can interpret as emergent. So open loop feed forward emergence, you know, a bunch of low level rules happen. And well, you know, we know complex things happen from simple rules being iterated. But we've been studying their behavior. We've been studying their various competencies. And it's you know, it's coming. There's a lot more. So I'm 100% with you. Like, this is why we make these synthetic things, because we want to understand the plasticity. We want to understand the space of the adjacent possible and the bigger space that comes from, right? What is the, you know, the morphospace, both of behavior, of physiology and morphology for these things. So yeah, I agree completely. The one thing that I want to say, like everything that you've said, I'm on board with everything that you've said, but there are two things that I tend to focus on in our work. One is this idea that, you know, I think we should be really careful not to confuse the formal models that we have with the actual thing. That is, right? If you know, I'm not worried about Turing machine models of the mind or any of that, because I don't. They're formal models. They don't necessarily capture everything that's of interest. There may be some use, you know, for something in cognition, but there's no reason to say that a brain or a human or a living thing is a Turing machine. I don't think anything is a Turing machine. I think we have these formal models that capture some slice of what we're looking at. I think all these wranglings about what it really are misguided. I don't think anything really is anything. I think we as observers have different formal models that we use to, you know, they're lenses through which we see these things. Some of them are better than others. Some of them are more useful in some contexts than others, right? And I don't think we need to, you know, worry about what it really is. I don't think any of these things really is anything. And where that really comes into play is, and this is some of our latest stuff on simple algorithms that we've been doing. For the same reason that organicists look at living things and say, look, your models of you know, your formal models of chemistry and you know, and all these things are not sufficient to capture all of the, you know, the beauty and the capabilities and everything of life, right? For the exact same reason, we should be very careful to say that our formal models of simple things, right? So whether they be sorting algorithms like we study or Turing machines or anything else, that those things fully and completely explain what simple things do. I don't believe any of that. So I think all of these formal models have limitations. We see these limitations as early as dumb sorting algorithms. We already see these things doing something that is not in the algorithm. And this is a paper, the preprint's been up for a while, the real paper will come out in a couple of weeks. I think very early on in the spectrum of complexity, you already get an insufficiency of these formal models that we use. Can you look at something as if it were an algorithm? You can, and then that'll help you do certain things. But I don't think we should think that, okay, well, now we've sort of plumbed its depths and that's all that it is. So that's why I really don't get, you know, too worked up about, you know, Turing machines or any of that, because it's one lens. Sometimes it helps, but I don't think it gives you everything you want to know about these things.
So I think it's lovely. So one of the things, so among things to contemplate, then I'd like to go someplace that's close to your Anthrobots, I think, but it's more articles where people discuss beyond the formal world. Okay, what does it mean to be beyond the formal? We're still deeply in the Newtonian paradigm. So put that on the side. So Kate knows all of this. This is slightly in confidence, but not really, Michael. So I'm working with this wonderful guy you'll have to meet. It's our friend Jan Dijksterhuis. Jan Dijksterhuis lives on a house on a dike, as you can tell from dike to dike. So 2 years ago, we came up with the 140 species experiment, and we have money in it. We're doing it in Holland. And it's getting neater and neater and neater. So we now have 70 fungal species. Each is DNA identified and tagged at the genus level, at least. And we have 70 bacterial species, each DNA sequenced, and each is stored separately right now at -80 or whatever it is. Okay. Our plan is within months now, it's taken a long time to get here, to mix these equal molar, equal number of each kind of species. So we're going to have 140 species that have never seen one another before, and plate them onto some sterilized dune sand, like maybe 50 times onto a meter square, and watch them for 2 years. And so the question is, so what do they do? Nobody's ever done this experiment, right? It's sort of like your Xenobot experiment.
Yep.
It's nuts.
Yep.
Okay.
Yep.
Nobody's
Very
ever
cool.
taken 140 species and said, so what are you going to do with one another?
Yeah. Yeah. Yeah.
So with one another. So from the third transition, we can't deduce what they're going to do. Step one. Step two, if pre-adaptations arise, mutations will arise over that time course. So if we take 20 copies of the same system on 20 plots, do the same mutations arise or do different ones? Well, probably different ones. Can we see that a molecule that used to have one function has some other function, which we can ask in the Xenobots also? Meanwhile, Jan has an absolutely lovely idea that I'm now in love with. His ideas of villages. So we put down this 1 meter square 20 or 50 times, and his guess is we're going to find little nodules in the soil, maybe a couple millimeter radius, and it's going to have some subset of the 140 species in it, say a specific 7 bacteria and 11 fungi. Call that village 1. How many times does village 1 occur? Now, maybe we'll find village 2 that has 18 bacteria and 4 fungi. How many villages are there? What's the size distribution of villages? You know, are they small, like 5 and 10 things, or are they huge? Do different copies of the same village accumulate the same mutations? We have no idea. Meanwhile, we are now in love with it. You'll like it right away. I realized about a year ago, there's nothing special about catalysis. It just speeds things up. So my example now is I have a flat tummy and you and I are bacteria. You can crawl over me to get food. I've afforded you a way to get to food, and an affordance is the same thing as an enzyme. It just lets something happen faster. So instead of my beloved idea of collectively autocatalytic sets, it's a set of things that are a mutual affordance set or a collective affordance set. My hope is the villages that Jan is imagining are collective affordance sets. Then we can ask of such a collective affordance. It's roughly, how do you guys make a living with one another? And that's obviously what's also going on in your Xenobots and your Anthrobots.
Yeah.
I mean, it has to be.
Yeah.
Then we can ask, is it easy for this to arrive, Michael? And when it arises, are molecules and properties being used for their old functions or wacky new ones that we could never have said? So is this idea of life will find a way truly generic? And are we seeing it, but we don't know to look for it, but it's been there all the time?
Yeah.
You're like, why didn't life vanish once it emerged? Well, maybe if you have enough species, it can always reconstitute itself. And there's something so beyond formalization that's emergent and collective, and so I love villages.
Yeah. No, that's tremendous. That's very cool. And I think, I think one of the things that we need to do with formalization is to remember that formalization assumes one observer that is trying to formalize. And I think what's very different in biology is, and this is a concept of polycomputing that Josh Bongard and I have been developing, that what I think you have in any kind of a biological system is an incredibly rich soup. It's a multi-scale soup of observers that are constantly trying to interpret and hack each other. So the interpretation, the models that they're making of each other, how they interpret the signals, it's a very pluralistic kind of vision, because they're doing their best. Every system is doing its best to understand what are these other systems doing. It makes these models, and there isn't one unique, objective, privileged one that is correct. Everything is up for interpretation. And in the entropy paper that I just put out a few weeks ago, it's what you just said is really important. Not only do Xenobots and synthetics and the kind of synthetic chimera, I mean, you're putting together a chimeric population system. Not only those kinds of things have to handle novelty, but I actually think every normal embryo and every normal standard biological system is figuring out what's going on from scratch every single time. I do not think that any of these things overtrain on the past. I don't think they take the past too seriously in the sense of here are my genetic affordances, here is my environment, I know exactly what it means and it's fixed. I think they reinterpret this stuff on the fly every time. Now, it just so happens that with the same starting conditions, they end up doing the same thing, which gives us this idea that development is reliable, it's fixed, it's this is what they know how to do, is this standard embryo. But the reason that we see these amazing examples of plasticity where we can put an eye on the butt of a tadpole and they can still see out of that eye, and we can move scramble the face and they all come back to where they need to be, all of these things work, these novel things, I think, because the biology never expected anything to have a fixed meaning, not the genes, not the signaling factors, none of it. And this is what we're working on now on the evolutionary scale, but also on the cognitive scale. Because if you think about it, at any given moment of life, you don't have access to the past. What you have access to is the engrams, the memory traces that your past self has left for you. They're messages. They're messages from your past self. Now, when you get these messages, you don't necessarily know what they meant. You have to reinterpret them, which gives rise to confabulation and construction, active construction of the cognitive self, and all this kind of stuff. So I think all of biology. I don't think this showed up when brains evolved. I think all of biology is doing this all the time. I live forward. I don't live backward. I need to figure out what is a coherent story about my environment and myself that I can tell right now based on whatever information I got from my past self. I don't care what my past self meant by it. My job is to reinterpret it right now in the most adaptive way possible so that I know what to do next. And that aspect, this idea that biology doesn't optimize for fidelity of information the way that our computational devices do, right? Like all of our computing devices, you better believe your bits you need to stay what they were before, right? You can't have them floating around. But I think biology assumes that the medium is unreliable, and it is not tied to one particular interpretation. Everything's going to change. Not only your environment's going to change, but your own parts are going to change. They're going to mutate. They're going to evolve. You have no idea. This is why you can do things like, if you imagine a salamander, right? You can very early on, you can make them have multiple copies of their genome, so they're polyploid newts. They end up with very large cells. The actual salamander is still the same size, so they use fewer bigger cells to build the same structures. And if you make the cells really gigantic, things that normally have 8 to 10 cells in a circle in a kidney lumen, they will take one cell, will wrap around itself, leave a hole in the middle, and give you the same lumen. Completely different molecular. Completely different molecular mechanism, right? No cell-to-cell communication. There's a cytoskeletal bending. One cell bends around itself. Now look, you're a salamander coming into this world. What can you count on? You can't count on having the right number of chromosomes. You can't count on having the right cell size. You can't count on having the right cell number. You need to have an algorithm that is going to figure out some way. And if you're completely messed up, you might be a Xenobot. You might not make it to be a salamander, but you're gonna have, you're going to be able to put yourself together despite incredible variety. You can't count on any of that stuff. And that's why, both cognitively and I think it's the same problem. I think all of this is cognition, like all of it, and at just at different scales. And I think that's what it is. It's uncertainty and it's reinterpretation on the fly. And there is no fixed model. Everybody's doing the best they can and reinterpreting the information that they have.
I'm just loving this. Let me go in two directions. Second direction will be to come back to Kate, because the confabulation is making sense of your world willy-nilly. By God, somehow it's going to hang together. And the criteria is the whole hangs together somehow. But also this gets at whether or not a living organism is at any moment something you might call a machine. So let's just take a typical machine, a cannon or a meat grinder or whatever it is. The parts do the same thing over and over and over again. And they do. It's a metal meat grinder, and the parts do the same thing. There's no reinterpreting of anything. Okay. In that sense, it's like a Turing machine. The cogs on the wheel turn the cogs on the other wheel, and the millstone grinds slowly. I think what you're saying is we're really trying to say the same thing, that at any moment, the causal properties in an organism work together, and the causal features that get used at any moment might keep varying in all kinds of weird ways. So you get this huge polyploid cell that makes something that's it builds a hole through itself. So it's still doing a liver membrane or whatever it was thing. In other words, maybe these are all stories of life will find a way, of a sufficiency of jury rigging, a way to make sense of it all in some holistic way. And Kate, it seems to me that this comes back so strongly to your notion of it's something like emotional coherence. Somehow there's a word coherence here, which is like the Kantian whole, which is like the Xenobot managing to do something, getting on with being a Xenobot, that's totally missing from our physics. It's totally missing from the machine metaphor of clockworks. So you try it, Kate. What do you think?
Oh, well, I've been trying to really create like the biggest picture, the simplest picture that can be grounded in direct experience. And of course, I think that understanding the function of emotion is something that's going to change. It reverberates down through biology and even physics, and then of course all the way up into social stuff. But that in terms of I've been thinking about just calling, all right, we need to identify the agent in the machine, because all the way up and all the way down, there is, as Michael suggests, this plasticity and this on the fly creativity in the moment that's adaptive and novel, and it's part of how life works at such a deep level. That to me is what's the very most important. And it seems like in almost all the conversations we're having, we're working with a quantum physicist to kind of bridge psyche, biology, and physics. And it's all coming down to what the structure of a Kantian whole is. And we need to kind of clarify something that's happening at every level. And to me, the way to do that is to think about boundaries, internal and external, up and down. And that, to me, it makes the most sense to think about internal and external on every scale, because external is going to be like from the top down, the outside in. But it's really the outside in. So the fundamental boundary between the self and its environment, whatever that self might be. But I think the concept is really important, because a Kantian whole is an identity structure, and that you have activity, flexible activity, decision-making toward two purposes at every level. One of them is to retain the stability of the organization and the order of the internal, and to adapt to the chaos and the requirements of the external world. So you have a self, not self boundary that's built into the story. And we're working on talking about that, defining that boundary as part of this dance between res potentia and res extensa, and the role that consciousness and intelligence plays. And I've been interested in people like Don Hoffman and how he's talking about
Oh, Mike talked to Don Hoffman, you remember?
Well, Mike has the coolest conversations with everybody, and it's really an honor to have access to this and to participate in it. But that the idea is that at this nexus of what we mean by a Kantian whole. There is a bottom-up kind of an amplifying feedback going on and a top-down homeostatic kind of feedback, or that's the closure that gives you constraint closure, organizational closure. It's called all kinds of different things, but it's actually information closure, because there's something that's being amplified, but that you see there's the parts are doing the same thing as the wholes in their environment, and that at every level you have both of that going on. So what in the paper that I wrote about this in 2014, I used the chemistry of the E. coli bacterium and infotaxes, if you will, as an example of, hey, this is what creatures can do, and this is what early creatures must have been doing all along, to argue that there is sentience and intelligence that's always played out in the evolutionary process on here and now timescales, but that but all I could really use was the cybernetic feedback model, and it's great because it fits what I'm saying and what flows upward in terms of the behavior of organisms. But that then Mike's work came out and I realized, oh my God, these feedback dynamics and the circuitry, this is bioelectrics. So there's this language, if you will, that needs that Mike's got his thumb on the pulse of, that can link this missing piece of the story to the network dynamics of system science, the dance of parts and wholes. And what I see happening is, I think I mentioned this once before, Richard Watson's work with song. The idea of resonance is one way. It's probably one of the best ways, I think, to get from sort of the physics up to any kind of sentient experience, any kind of embodied way of harmonizing with the physics. And what I see going on with Mike's work, and I'd love to just chat with you alone about this sometime to straighten me out about it, but that the cell membrane at resting potential seems to be the associated with some sense of home state, like I like to say it's the edge of chaos state, because the cell knows that's where it wants to be. It's the preference in the system. It's the right place to be. It's like least energy or whatever story it's hooked into, but that there's an awareness of that and an urge to maintain that, get back to that, because you're being constantly tossed off of that with energy exchange and everything else, and connecting in networks is even, so that's the right state, but it's not just a state of an individual. There's two aspects of identity. It's the whole that always needs to be on the edge of chaos, but it's also the part that needs to be in a collective with others in the same space, such that they can all interact and do something communally. So I see that as the harmonics where we can sync together in a collective way. And Richard Watson's really thought about this carefully, and he's talking literally about simple binary motions that have to do with quantum mechanics and rotations, geometric rotations and trajectories and all of that, where just the tiniest little bistable rotation, you can literally see a whole bunch more of something that's close to self, the nearest neighborhood, your nearest neighbors, and you can sync up with them through the harmonic resonance. So what I was describing in my paper as this three-step loop, where there's an ongoing comparison between the self and the not self environment, that what I see from Mike's work is happening at the cell membrane, and the use of the bioelectric polarity, going like more polar, is associated with the kind of more free energy that I can use, dynamic that I call self-development, and the lack of polarization is associated with the less, don't let this happen kind of situation. But it's really about plasticity. And I hope you're following me on that, but
Oh, yeah.
I'll get some of this written down better, but that I think it's really the algorithmic bit of it. We need to clarify the machine, because there is like a bistable switching thing happening that's common, that allows this connectivity all the way up. And I think that the edge of chaos story and the idea of Stu's original idea of Boolean networks and all that is going to be central to cracking that algorithmic code. And I think it's the edge of chaos. So that's a little bit of a splatter.
Yeah, yeah. I know that's super interesting. I mean, the thing about bioelectricity, and no one loves bioelectricity more than I do, but one thing about bioelectricity is that it is really good at showing you how selves and their goals, which I take to be very closely related things, scale up. So as you point out, from the homeostatic properties of a single cell bioelectric pattern, we can show, now we see quite clearly how these things can, when the cells join into larger scale electrical networks, what happens is that the size of the goals that they can pursue not only enlarges, so that cognitive light cone gets bigger, but it gets projected into new spaces. So for example, whereas a single cell was only concerned with metabolic goals, physiological goals, bioelectric goals, whatever, the collective is now concerned with morphogenetic goals. And hey, do we have a proper limb? And if we don't, let's build more until we do. And right, so the bioelectrics is great at showing how minds can scale. That's one reason why we study it so much, is because it shows in a very clear, molecularly realistic way how the goal states of systems can enlarge and project into new spaces. But having said all that, the interesting things that we care about in terms of living things and I would say some non-living things, but cognition, the thing, it shows up way earlier than that. So even so, for example, we've shown that if you look at it the right way, and this is like what Stu was saying before, you could think of it as a paperweight, and then you'll miss a bunch of stuff. If you look at it the correct way, even very simple gene regulatory networks, we're talking, 5 nodes and up. If you look at them the right way, they can do 6 different kinds of learning. They can do habituation, association. They can do Pavlovian conditioning, right? You can, and you don't need the rest of the cell. You don't need a nucleus. You don't need a bioelectricity. You don't need a membrane. You don't need any of it. Just from having certain specific relationships among the nodes, you get no, you don't need transcription, you don't need promoters, you don't need any of it. Just a small number of molecules that turn each other on and off, you can already get. So some aspects of what we like about cognition, the ability to learn from experience, and it starts up very, very early on. And the other thing that starts up very early on is something else that Stu was talking about, which is the ability to do things that were not explicitly in the algorithm. So this creativity, and Stu, when you called it life finds a way, which I think is brilliant. In my paper, I called it beginner's mind. Same kind of idea, right? That you are not tied to some fixed interpretation of what's going on. You are an agent whose job it is to, and whose opportunity it is to figure out how to deal with new affordance. What can I make of this, right? That's the basics of intelligence. Use the things you have in some new way. I think that starts extremely early on the scale. I don't think it requires even cells necessarily. I think that very simple mechanisms in their own simplified way already exhibit this ability to do more things than is apparent to us if we use an algorithmic lens on them. And so this thing with machines, so here's my take on it. I think that if we think that these are binary categories, you got machines and then you got living things that are not machines. I think we're in huge trouble there. What I like is a spectrum. And I think that it's perfectly fine if your orthopedic surgeon wants to see you as a machine. That's cool, because he's got hammers and chisels, and he better, that is the frame that they're using, and that's going to be okay for some things. If your psychotherapist thinks you're a simple machine, that's no good. So there are different lenses that you can take on different things. For some, they will be useful. Is a meat grinder kind of, as you said, machine useful for biology? Not really. But there are other aspects of the machine metaphor that are of different machine metaphors that are useful for other things. They do not capture the whole thing in any way. But I don't think we should have these binary categories where we say it really is a machine or it really isn't a machine. I don't believe in any of that. I think these are all frames that we take on, and then the beauty of it is that in the empirical testing we can compare. You can bring your metaphor, I bring mine, and we say, what does this help you invent? Right? Did you make Xenobots? Did you make Anthrobots? Did you make some sort of regenerative intervention? Well, no, because I was using a bottom-up machine metaphor where molecular rules percolate up to complexity. Well, I did because I was using a top-down controller where I thought the cells actually have the ability to solve certain problems, and I've manipulated them to solve it in a different way. So there it is. So that's what I think about these metaphors. And the other thing I wanted to come back to a little bit was about this business of deductive and inductive. So if you this is something that I was trying to lay down in this latest entropy paper. If you think about the process of let's just go back to the butterfly caterpillar thing, right? So there are memories in biology. There are cases where memories survive drastic remodeling of the brain. Not only do they survive, much like any species, right? A species always has this paradox. If I don't change, I'm going to die out because the environment will change and then I'll die out. Or I can change, but then I'm not the same, so then I'm gone again, right? You have this paradox. So memories have the same thing, because if you're a caterpillar memory, not only in order for you to survive into a butterfly doesn't, you learned about associating leaves with certain color stimuli, and you were trained for this memory. Those exact memories are of no use to the butterfly whatsoever. Butterfly doesn't care about leaves, it flies, it doesn't crawl, everything is different. For you to survive, you have to change. And what happens is that if you think about, think about learning, right? So you have all these distinct experiences. They get squeezed into, by a process of generalization, they get squeezed into a simple rule. Some n-gram that you've got. The particulars. You've learned a pattern, right? And even algae can learn patterns. We got a paper coming out next week showing surprise in algae, right? They get surprised because they have expectations because of patterns. It's algae. So what happens is
that's the title of the book, The Surprised Algae.
The Surprised Algae.
Yeah.
Yeah, who would think? But they can be surprised because they catch on to patterns, and then they have expectations. And so you squeeze your experiences down into, right? So you think I visualize this as a kind of bow tie, like the middle of an autoencoder, where you take all these experiences, you squeeze them down into a very compressed representation because you've gotten rid of all the correlations. But and so that's your past. So your past self has done that. It has left for you some n-grams, but now your current self has to reinflate those. And you can't do it deductively because you're missing information. You don't have all the information where you can just know exactly what it means. That's the whole point of the compression, is you've gotten rid of all of that stuff. So now you have this thing, and now you have to say, what the hell does this mean? What does this memory mean? And if your brain stays exactly the same and everything else stays exactly the same, then your interpretation is likely to be what your past self's interpretation was. But it doesn't have to be, and you have no guarantee that it will. And so that becomes a creative process. That is now very much a creative process, because you cannot deduce what it meant because you don't have all the details. And so I agree completely. I think these deductive metaphors only take us so far. They're useful for some things, but they only take us so far, because the fundamental thing that you see in evolution, in development, and in cognition, which I think all of these are cognition, is this creative interpretation of the stuff around of affordances. And your memories are also your own affordances. And you are handed these molecular or biophysical, whatever they are, you're handed these things. You need to figure out how to use them, and you are not necessarily tied to how your past self used them. So I completely agree with you about this deductive inductive thing. Creativity and problem solving is at every level. And so, as you said, Kate, in the homeostatic bioelectrics, but even below that and above that, like it permeates everywhere.
Can I just make one quick comment? So all of the information is getting compressed down. I love your bow tie, by the way, and it fits with the Dow story. But that what would you say is the very, very most important compact information that is then stored that then can be reinflated? And does it include any kind of value system?
I think it must, because if you think about okay, so you train the caterpillar on some color cue, and then here are the leaves. So the caterpillar learns, I crawl this way and then I get these leaves. The butterfly doesn't care about leaves, it wants nectar. And so one of the things it has to remember is not, hey, I got leaves, maybe not even I got food, maybe it's, this was really nice, I enjoyed that. And so, right, so how much generalization, we don't actually know in that particular case, but this is something we can study. And by the way, we're studying memory in Anthrobots, because we're gonna check if you can recover some of the donors, the human donors' memory from these Anthrobots. That's a whole other thing. But that's
cool.
right. But that squeeze down is, maybe the valence of the experience is critical about, right, about, I mean, what's the first thing you need to know about any kind of procedural memory like that is, was that good? Was that a good thing I did, or was that a terrible thing I did? So that's right, that's the core of it. And then after that, you can layer on top of, well, yes, it was good and it was delicious, or yes, it was good and something else happened. But no, I think that's, and I don't think, that's another thing, is the memory, like a lot of people think about what's the memory mechanism, like where's the encoding, right? What are the engrams? At this point, I suspect that there is no single memory medium. I think that basically what's happening is that, and this is a total conjecture at this point, but we're doing some work on this. I think that what's happening is that all of the levels, the molecules, the cytoskeleton, the signaling pathways, the lipids of the memory, all of it is being used as a reservoir, in the sense of reservoir computing. And what the neurons are doing is interpreting the patterns in the reservoir. I think the memory, it's like there is no single memory mechanism. It's just incredibly opportunistic, and it uses whatever scratch pads it can. And then future you is good at reading those and trying to make a coherent story out of it. And so what the nervous system is doing is more interpretation than it is actual storage. That's a very contentious, the whole like synaptic, memory stored in synapse is very contentious now. There's many cracks in that story. So, but yeah, I think you're right. I think that kind of, the emotional valence is probably key to these things.
Yeah, I just want to say that that's why I've been suggesting that there's a fundamental semantic information bit that is binary. And it does give that yes, no value system about no to anything that's going to destroy the structure and the stability, and yes to everything that's going to be a novel, new, adaptive opportunity. And that those two things come from the basic sense of, am I in my optimal state? Yes. And am I able to connect with others in, so there's a dual memory here. I mean, not a dual memory, but a dual sense part identity structure where I am at once whole and balanced as an individual, but I am also whole and balanced within a collection. So you have a social aspect, a collective aspect, and an autonomous aspect of identity all the way down.
Yeah. I mean, the only thing, so I agree with all of that. There's one layer on that I think we need to think about, which is that as all of us change, the valence is going to can change too, because for example, think about the metamorphosis from, I don't know, you're a dragonfly larvae and you're going to become a dragonfly. So when you're a dragonfly larva, you love being underwater. Yeah, you love being in the water. When you're a dragonfly, you don't like that at all. So one of the things that has to happen under certain changes is that valence, may also have to adapt to your new self. You can't just leave.
The valence itself isn't adapting, but the memory of it is. And it's because of the valence that you can have those memory categories. This is straight Pavlovian conditioning, in that you get extinction after a while, but it's the fundamental binary information that stays the same.
Well, I just mean that if you have a memory that something was good for you, by the time you retrieve it and you now have changed, you're a different being than you were before, you're going to have to reinterpret. You can't just hold on to, yes, this was good, because because by now it may no longer be good, right? So it can't be constant. You can't hold all those things fixed as you change.
Well, it's not. That's why pain tells us, hey, this isn't working anymore. I mean, that's the reality sandwich of the fact that you're no longer the same. I mean, that's the way cognition actually works. And that the substrate of emotion is a different process, and they work together. And that's where some of the cognitive science, I think, can be misleading, because you've got to make that distinction. Emotion is always from the embodiment. And there are specific things that the embodiment needs. And of course, when you have something as beautiful and dramatic as metamorphosis, those things are going to change. But that's a rare kind of, yeah, I mean, sure, I want to understand.
Is it rare though? Is it rare though? Because I mean, okay, in that drastic example, it's rare. But all of us were kids once, and our preferences changed pretty drastically, right, across puberty and whatever. Things that seemed really important don't, and vice versa. I think we all have a smaller version of that. We're all changing fairly continuously.
Well, this is where you get the physiological needs of the body as the basis, sort of universal internal goal states and needs. But then, and that's what the basic emotions kind of help us understand all through the lived. Certain things like agency and autonomy and community are all necessary. But if community costs you your autonomy and your freedom, you break away from it in that moment. So these things are mediated by the self-regulatory information that emotion provides. And once you kind of get that, it weaves together and it gives you a better understanding of a whole bunch of literature that's out there concerning the self, concerning motivations, concerning how other people can get into your OODA loop by providing rewards and punishments that engage your bodily responses with your mind out of the loop. It's there's a lot more going on there, but what we're talking about is the very, very beginning of whatever algorithmic aspect of the machinery. Because when I say machine, I'm talking about the fact that there are certain patterns of living systems that have evolved, that are emergent of laws and forces of nature. And I'm not at all sure that bioelectricity doesn't go all the way down in terms of quantum biology. In fact, I think it does, but that there clearly are things that are going to happen that are deterministic, whether we like it or not. And like, I'm going to drop my pen and I know it's going to fall, but that one of the things that is part of that determinism that we are not creating is when stuff goes wrong, we feel pain. We don't like it. And if we were constructing everything, we wouldn't have any need for that. Right? So it's a reality sandwich that's incredibly important.
Yeah. We're working now on some things around rewards and punishments for gene regulatory networks. How do you punish a GRN? Well, there better be a way, because you can do it with a paramecium, and that thing's chock full of GRNs. So right, so trying to figure out like the very kind of the very basement of it.
I'm so glad you're doing this work. I mean, it's wonderful. It's just music to my heart that you are understanding the importance of what you're doing and making so much and so many connections for so many of us. Thank you. Go ahead.
Thank you.
What's the paper you've just published at Entropy, Mike?
I'm gonna send it to you. It's called Agential Memories as a Cognitive Glue. I'll send it along.
Yeah. So meanwhile, I have written I've actually been doing, of course, some cosmology, because every Jewish fruit fly geneticist should do something. So it's a long, long, weird story. A new colleague in Botswana, and I think we know what dark matter and dark energy is. And if we're right, it's a creation of space-time by matter. And that's another story, but it's really weird. Meanwhile, I published this paper with Sudeep Patra in India with the outrageous title of Cosmos, Mind and Matter, : is mind in space-time? Mike, it might be right. It is really weird. So it's online. It has been accepted for publication by Biosystems. It's out, except that all the references are screwed up, but it should be straightened out in a few days.
Awesome.
And I'd actually kind of like to send it to you. It's very,
Please do. Yeah, please do.
it's
Well,
really,
they're working on a project too of the evolution of free will that gets at exactly these issues about what a Kantian whole and all of that. So hopefully we'll be able to put something together.